I'm going to stop sharing, and we can now move on with the first session of today, entitled Illuminating the Tree of Life. Our first speaker is Anja Spang from the Royal Netherlands Institute for Sea Research. I have to say that Anja was our selected speaker for the meeting in the Society for Molecular Biology and Evolution that never happened. And we are really grateful to her for accepting our invitation for being here today in this format. He's going to talk about the exploration of the archaeal domain of life as a window into key transitions in life's evolution. Anja, welcome, and please feel free to unmute yourself and share your video when you're ready.
Yeah. Hello, everybody. Can you hear me? Okay, perfect. Then I'm just sharing my screen. Do you see my screen?
Right now we are just like seeing a picture of you, but not like share the screen so far.
You should be able to share now. Please give it another try.
I'm sharing it. Can you see anything? No? Okay.
Can you stop sharing for a sec and then try it again?
Yeah. Trying again.
There we go.
Yeah.
We can see it now.
Okay, perfect. And I guess I play my presentation. You still see the slide. Is it now full screen? Perfect. Okay. Thank you very much, Rosa, for organizing this virtual conference. I'm very happy to present here today, bringing you into a little bit into the world of the Archaea as a window into the early transitions of life's evolution. And so you all know that Earth is a diverse world of microbes. And what fascinates me about this is that all life is connected by common descent. And this is easily seen by this famous tree that you probably have all seen many times by Carl Woese in 1990, that also led to the discovery of the Archaea as a separate domain of life besides the bacteria and eukaryotes. And what was very early on shown in this tree was that Archaea seem to share a common relationship with eukaryotes to the exclusion to bacteria. And that made Archaea very interesting for understanding evolution and the earliest stages of cellular complexity in eukaryotes. However, during the past years, metagenomic sequencing has enabled a completely new perspective on life's diversity. Here you can see the famous tree by Laura Hack that was published in 2016, and that includes not only cultivated organisms, but also the vast diversity of uncultivated microbes that are out there in the environment, but for which we know nothing but their genome sequences. And so what this tree, among others, has shown is that there are two major lineages, both in the bacteria and in the Archaea, that have not been seen before and that are comprised of very small cells. So they are here on one side, the CPR, the candidate phyla radiation, and on the other side, the DPANN Archaea. And these organisms seemingly branch deep in this tree of life, and therefore are thought to perhaps provide new insights into the early evolution of life in general. However, there is one problem because we actually don't know where's the root in the tree of life at this moment. Is it between Archaea and bacteria, or is it somewhere inside of the bacteria? Furthermore, we don't know if this is actually an artifact that the deep branching DPANN are not really deep branching. So why do we think this? So the DPANN Archaea, for this we need to first understand who they are, what they are. And so the DPANN Archaea, they have very small genomes and limited metabolic potential. The cultivated DPANN that we know of have predominantly symbiotic or parasitic lifestyles, and that is now here illustrated by interactions between Nanoarchaeum equitans, which is a parasitic symbiont of Ignicoccus hospitalis, the big cell on the left hand picture, and then also by nanoHaloarchaeum, which was recently cultivated and shown to be a ectoparasite of Halorubrum lacus profundus. So that illustrates that these organisms, they are symbionts, at least most of them. And so the problem with symbionts is that they often have compositional biases in their genome sequences, but also in their protein sequences, which can lead to long branch attraction artifacts in phylogenetic analysis. And so you can see here these two trees, and the question is whether the tree on the left side is an artifact where the DPANN branch deep in the tree due to long branch attraction to the bacterial outgroup. And so our aim was to resolve the DPANN placement in the tree of life and provide insights into that diversity. And for this, we first looked, what do we actually have out there in the databases? And so we reconstructed a small tree using, or a tree of just a few ribosomal proteins to get a first view of the diversity that we have in metagenomes. And this shows that there are about 11 different DPANN lineages known out there that are on top of the tree. And so they can be broadly divided into two clusters, cluster 1 and cluster 2. And the interesting thing is that cluster 1 includes organisms that are free living and that have much larger genomes. But then there's also cluster 2, which comprises mainly symbiotic DPANN archaea, and also these examples that I've shown, nanoarchaeum and nanoHaloarchaeum. And then we also saw that there was one additional lineage that grouped between all the DPANN and all other archaea, which was referred to as the UEP2, the uncultivated archaeal phylum 2. And so we wanted to know who are these organisms and do they hold insights into the early evolution of the DPANN archaea. And so we screened the currently available metagenomes for sequences from these UAP2 organisms. And that allowed us to reconstruct and analyze new genomic bins. In total, we were able to recover 12 genomic bins of members of this group. And so we were asking, does the UEP2 clade really represent an early diverging DPANN lineage? And in turn, are all the DPANN early diverging? And so in order to address that, we used a careful phylogenetic approach that was supposed to help to reconstruct a reliable phylogeny of the archaea, which is not very easy, especially considering that these symbionts have compositional biases. But first of all, we started to identify suitable markers that we can use for protein concatenations, because of course we should only concatenate proteins that share similar evolutionary history. And to achieve that, we developed a new marker ranking scheme system where we had two major criteria. Both archaea and bacteria should be monophyletic clades in these single gene trees, and major archaeal clades should be monophyletic. And so just to illustrate an example, you don't need to see the details in this tree, but what you can see is that all lineages on top of this tree are belonging to the archaea, and in color you can see the different orders within the archaea that are conserved, and all of the archaea form a sister clade to all bacteria. So this is clearly a marker that has a conserved domain monophyly and is therefore suitable for phylogenetic analysis. However, we had also many of the universally used marker genes, including those from GTDB or PhyloSift. They included markers in which bacterial and archaeal monophyly was not at all conserved. So you can see here on the right-hand side that bacteria were intermingled with archaea, and clearly this marker should not be used in concatenations. Another example here, we just looked at the conservation of archaeal order level lineage monophyly. And so illustrating here with one clade, the Undinarchaeota, which forms a monophyletic clade in this marker protein in the top of the tree. And then another example of a single marker protein tree where the Undinarchaeota are actually all over the place, suggesting that the left-hand side marker is not a good phylogenetic marker. So what we did then is to to count this violation of monophyly across all the orders in the Archaea as so-called splits, and use this for ranking the markers. And subsequently, we performed phylogenetic analysis of just the 25% and 50% highest ranked markers and lowest ranked markers on the other side. And so we used all kinds of different complex models of evolution that best fitted our data in both maximum likelihood and Bayesian, and also performed various additional steps such as compositional side filtering and fast side filtering and so on. And so what was very interesting to see is that when we used the 25% lowest ranked markers, we recovered sisterhood relationships of hosts and symbiont clades. So you can see here on the right-hand side, we have in green, the nanoarchaeota are placed within the granarchaeota, and the halobacteria are a sister clade of the nanohaloarchaeota inside of the DPANN archaea, in spite of them being in fact euryarchaeota. So that clearly suggests that host symbiont transfer contributes conflicting signal to the archaeal phylogeny. Next, we also concatenated the best markers, and this is just an illustration of one of the many trees that we obtained, in which we concatenated 25% of the highest ranked markers, and that recovered a topology that we saw in most of the trees. And in all of these, the UAP2 represent a new archaeal phylum, which we named the Undinarchaeota. And it also shows that the Undinarchaeota are in fact not branching deep in the DPANN, but they form a sister clade of the DPANN cluster 2. And to remind you, the DPANN cluster 2 DPANN are those DPANN that have the smallest genomes and that are the most dependent on partner organisms. And furthermore, the Undinarchaeota form two order-level lineages, which we refer to as the Undinarchaeales and the NIOZ Archaeales. These are water nymphs after which they were named. And so we confirmed these results with a variety of different approaches, like fast site filtering and compositional biased site filtering, as well as taxon sampling. So once we obtained these three, we were wondering, okay, but what do the Undinarchaeal genomes tell us about their metabolism? Are they really symbionts? And so we reconstructed the central carbon metabolism and energy metabolism of the Undinarchaeota, and it showed that even though the Undinarchaeota encode nucleotide biosynthesis protein, they encode a pentose phosphate metabolism and also an AMP salvage pathway, which may allow them to conserve tiny amounts of energy. They have various gaps in their metabolism. For instance, they lack various pathways involved in anabolism, such as amino acid biosynthesis, vitamin biosynthesis, and also archaeal lipid biosynthesis. And they also lack a citrate cycle, which indicates that they have various auxotrophies. And so this suggests that they are likely dependent on partner organisms for the acquisition of certain metabolites. So we then also looked on a broader scale on the distribution of proteins involved in catabolism and anabolism across all archaeal lineages. And this shared again that the Undinarchaeota, which are highlighted with this violet bar at the bottom within the DPANN, share many features in common with the DPANN cluster 2 clades. And so most importantly, they all lack amino acid and vitamin biosynthesis proteins, and they have a variety of shared synapomorphies such as gene fusions and gains of proteins, for example, involved in informational processing machineries, which we suspect could even have something to do with the loss of many genes and genome streamlining processes. And they also seem to have in common the gain of proteins that potentially mediate their interactions. So as a result of this, we conclude that the Undinarchaeota likely represent symbiotic lineages similar to the DPANN cluster 2 members. And so finally, we used our highest ranked marker proteins for rooting the archaeal tree and determining whether the DPANN archaea really represent deep branching lineage. And so this outgroup free routing method that we used was based on non-reversible models in IQ3 that were only recently made available. And what this is important because in this case we prevent the attraction to a bacterial outgroup, which might lead to long branch attraction artifacts. And so these the results of this analysis excluded the root from within the TAC, Asgard, and Undinarchaeota, but it did not strongly resolve the root position among the DPANN archaea. And the two most common roots that we observed were between all DPANN and the rest of the archaea, or between cluster 1 DPANN and the rest of archaea. So,
1 minute.
Yeah, so together this suggests that DPANN symbionts may have evolved in parallel or, throughout archaeal evolution. So to conclude on my presentation, the DPANN archaea frequently exchange genes with their host, which contributes to conflicting signals in phylogenetic analysis and of concatenated marker proteins, but can actually help to identify symbiont host systems. I did not talk about this here, but we can actually use these horizontal gene transfer to make hypotheses on who are the hosts of uncultivated DPANN. We have identified suitable markers that help to resolve the archaeal phylogeny and showed that the UEP2 lineage represent a novel phylum, which we refer to as the Undinarchaeota, and that the Undinarchaeota are a sister lineage of the streamlined cluster 2 DPANN. And finally, our analysis suggests that the Undinarchaeota comprise putative symbionts that have shared characteristics with the genome-reduced members of the cluster 2 DPANN. And so our next steps are now to determine the ecological role of the Undinarchaeota and hunt for their interaction partners. We also want to use transfers and duplications to root the archaeal tree and maybe get a more certain root position, study the co-evolution of selected DPANN and their host, and shed light on the mechanisms that enable these interactions. And so please contact me if you are interested in any of these topics for a PhD or postdoc position. And so finally, I would like to thank all my collaboration partners and also my team at NIOZ with my PhD student Tara Mahendrarkaya and Nina Dombrowski, and all of you for listening. Thank you very much.
Thank you very much, Anja. It has been a fantastic talk. Now it's time for questions. We don't have much time, maybe just for one question. I remind you how to do that. We should put write down the questions in the Slack. So far, there is no question. I have one, just in case no one is still wants to make one. I am curious. Okay, there is one question. I have one question. How you choose the best market by phylogeny, Anja?
So we performed single protein phylogeny of all markers. And then for each, we counted how many times was the monophily of all the different orders violated. So if, for example, the VUS archaeota are split in 3 places across the tree, instead of being monophyletic, that would be counted as 3 splits. And so we normalized this by the amount of sequences that we had per order level lineage. And so this allowed us in the end to rank the markers based on the total amount of splits across the whole tree.
Fantastic. Thank you very much. I will write down my question to be replied afterwards. I think we should move on. So Rosa.
Thank you, Anja.